So it is what, the 2nd of July already? Amazing. And this is the last lecture for the exam number 4 over the nervous system. Reminder that this exam will be on Monday, up to Tuesday for some of the hybrid students who are listening. And this exam is going to cover the introduction to the nervous system, which was chapter 11. Chapter 12, which was the spinal cord chapter, which I'm going to finish the last few slides today and kind of ramp us up into that. And then the brain, chapter 13. I know it says 14 here, but I'm off by 1. Chapter 13 in your Martini book. And then I've already pre-recorded the end of the brain lecture specifically over the cranial nerves. It's about a 15-minute lecture. It's on YouTube. I had originally recorded them separately, merged them together, and last night when I looked at them, one of them was all gray for some reason. So I redid them, and they're reposted. There's about a 15-minute, though, video over the cranial nerves, and then there's also about a 30-minute video over the autonomic nervous system and all that you need to know from what is chapter 14 in your Martini. Okay, so that material, introduction to the nervous system, parts of the neuron, overall structures, Schwann cells, ependymal cells, astrocytes, that whole conversation was the introductory chapter. And then the spinal cord structures and everything about the spinal cord. Today we'll go over the brain. And then your job will be to review the cranial nerves, the last part of this, and the autonomic nervous system, collectively about 45 minutes. And that content will be on the exam. That exam will be a full-length exam on Monday, and you'll need a Scantron 884-E, the one you've been using every time. And that will be exam number 4, and vocab on that exam goes, what, 51 through 70? So let me start off by going through the last little bit of vocabulary. Then we'll jump back to the last bit of the spinal cord, and then move into this chapter on the brain.
The link that you sent through the email, is that still good then, even though you broke them up?
I believe so. If not, go to YouTube. Good question. I did send a link out, and now I can't remember the order in which I sent that link. That link could have been to the dead one that I took off. If it doesn't work, and I apologize, I don't recall, just go into YouTube like you always do on Blackboard. It shows you, right? And then there's 2 short links, two links to 2 short videos. So vocab here. There we are. So we went through OV and OO last time. So through phleb on this exam. So let's get to 64. That is OP, like optics. OP meaning vision. Ophthalm, also relating to the eye. Ophthalmology, the study of the eye. Opsy, the viewing of something. So microscopy is the viewing of small things using a microscope. Biopsy, the viewing of living things, right? So we take a biopsy to look at living tissue. And necropsy, the viewing of dead tissue or of a dead body, not a human. So we reserve the word autopsy because auto means what? Self, right? So autopsy is the viewing of self, of our own species, if you will. And we would do instead of necropsy on a horse or a dog. Or oral, thinking about the mouth, oral cavity. Orbi, you've seen the orbicularis oculi, the orbicularis oris, or orbit, think about something that's circular. Orchid, we think of beautiful flowers, but it's also the testis. So orchiditis would be an inflammation of the testes. Ortho, you go to the orthodontist to get your teeth straightened, so straight or corrected or normal. And ori, pertaining to, so sensory pertaining to the senses. Oscopy, again, viewing. Lapar, remember, means abdomen. So laparoscopy would be viewing of the abdominal cavity. If somebody is very verbose, they are full of words, right? So os, full of, adipose, fat. Osis, a condition of, osteoporosis, a condition where the bones become more porous. And osmo, remember, osmosis was the movement of water across the membrane, and literally it's being, quote, pushed by the osmotic pressure. So osmo referring to push. Osteo, we've seen this one a lot. Osteo referring to the bone. Ostomy, formation of an opening. So tracheostomy, the opening of the trachea to restore the airflow. Odo, if something is for the ear, It'll say that it's like an otic drops, right? Drops for the ear. Otogenic, something that would be originating in or with the ear. Papillae, we're going to see this for papillae. We're going to see this term a lot in many different places. In the digestive system, in the fetal pig, or on the models, I should say, there was the duodenal papilla. It was listed in your Amerman book as the hepatopancreatic ampulla, but it was that little nipple-like structure where both the pancreatic juices and the bile from the gallbladder and the liver dumped into the duodenum. So hepatopancreatic, liver and pancreas, or duodenal papilla, this nipple-like structure where everything dripped in. We're going to see, though, in skin we saw the papillary layer of the dermis, little nipple-like peaks and valleys, and we're going to see a lot of papillary structures in different places, in the kidney and the heart. Otomy to cut, a lobotomy to cut out a lobe of a liver or maybe a lobe of a brain. Para, near or beside. Paranasal sinuses, those spaces next to your nose or beside your nose. Par, birth or labor. Postpartum, right, after birth. Parietal, we've seen this one long ago in Lab 1. We know the serous membranes are made up of two layers, the visceral and the parietal layer, and the parietal was lining the wall. Pathy, some disease. Pathology, the study of disease, or neuropathy, for example, a disease of the nerves or nervous system. And pecto, we've seen the pectoralis major referring to the chest. You go to a pediatrician for child-related things, so ped, children. Pelvis, your pelvis is a basin-like structure that's holding the reproductive and urinary structures. Penia is a deficiency in. When we get to blood, we'll talk about the leukocytes, the white blood cells. And so leukopenia would be a deficiency in white blood cell. Osteopenia would be a deficiency in bone. And penna, we saw that muscles have different arrangements, and some of them are described as being pennate. They have a feather-like arrangement where the muscle fibers are coming down on the tendon at an angle. And then finally for this test, like permeable, something can get across. Penetrate, or not penetrate, right? Just permeable, Peri, around, we've seen the perineurum, we've seen the perimesium, we've seen the periosteum, anything around. Phage, to eat. Phagocytosis, the cellular eating process. Macrophages are large eating cells. And finally, phleb, vein. If you're going to have your blood drawn, you're going to have it done hopefully by a phlebotomist, and we normally draw blood from the vein. Okay. So that is our vocab, 51 through 70, for this exam on Monday. The exam is largely multiple choice. There will be a case study or two. And what might you imagine? From what you've seen so far, what kind of case study would you anticipate reading about? Maybe something about myelin, right, and nervous system. That's not a bad idea. It's a really good idea, but it's not on there. But I like it. But you definitely want to know about myelin, right, and that myelin speeds up the conduction. And I did mention that MS is a disease where the myelin is defective, and so the conduction is slowed down. What else, though? Spinal cord injury, right? Likely someone's going to be in the ER, and they're going to have suffered some sort of potential damage to their spinal cord. And think about paraplegia and hemiplegia and that kind of thing. And today we're talking about the brain, so you can also anticipate a stroke. And we'll talk more about that today. So some basic understanding of what happens when there's brain damage, and that will make more sense to you as we continue with today's presentation. So someone's going to have likely some sort of head injury or stroke, and someone's likely going to have a potential spinal cord injury. And again, you'll read a little scenario and answer some questions, and nothing you really can study for, but just make sure you're thinking about those possibilities. The vocab will be fill in the blank. The case studies would be fill in the blank. And the connection questions would be fill in the blank. Don't forget there'll be connection questions. How does the nervous system connect to other systems, and what specifically is that interaction? So just think about, the brain and the spinal cord and the things that interact with it. It's not hard. Please don't leave it blank. I do cry tears on the way through to Grand Rapids sometimes, right about Coopersville. Tears start coming down, and I start banging on the steering wheel. When people put, I didn't study that, I can't think of anything. how the nervous system is connected to any other system. So please just take a moment, think about it, and then you won't have that excuse on the exam. You'll have easy points. So those are the fill-in-the-blank parts. The rest of it will be objective. And then at the end, someone say to me cranial nerves at the end, and I'll show you, I mentioned it in the pre-made video, but I will show you specifically again how I want you to study for those cranial nerves. And if there's time, hopefully there will be, I'll run through the cranial nerves with you so that you'll be that much further ahead when you listen to that video. Okay, so that's our vocab. Let's go back to the very end of the spinal cord. Now what I'm going to do right now is I'm going to zip through this PowerPoint really, really fast as a quick review. And if there's anything at all that I went over that you need to hear again or restated, please let me know. So let's start at the very beginning of this chapter on the spinal cord. And much of this was the structures, right? We talked about what the spinal cord is doing for us, and we went through the fact that the spinal cord ends around L1, that the spinal cord has two indentations on each side, those median sulcus or fissures. We know the different regions of the spinal cord, right? There's the cervical, the thoracic, the lumbar, the sacral regions, and the very, very tippy tip end would be considered the coccygeal region. From each of those regions of the spinal cord is derived the number of spinal nerves that we know. So from the cervical region, how many nerves? 8. From the thoracic region, 12 nerves pop off. From the lumbar region, there are 5. From the sacral, there are 5. And the coccygeal, there's just a single pair. That gives us our 31 pairs of nerves. The spinal cord comes to a pointed tip at about L1 called the conus medullaris, or the medullary cone, and everything inferior to that point collectively is referred to as the cauda equina. That is because it resembles a horse's tail. There is a strip of pia mater-like material that goes down the very center of this called the filum terminale, and that helps to anchor the conus medullaris down to the coccyx. There are wonderful structures surrounding and protecting the spinal cord. This would be not only the vertebra and the muscles in the skin of the back, but also the meninges. And remember that the meninges from outside in, the outer harder layer is the dura, the middle layer is the arachnoid, and the inner layer, shrink wrap to the surface of the spinal cord itself, is the pia. We see them located here. You've labeled them in lab as well. Now, this is where I want to take a moment and make sure we've got this. So, because we'll be talking about this also when we get to the brain. The central nervous system is oftentimes, we know it's the brain and it's the spinal cord. But you'll also hear people say white matter and gray matter. And what you're basically going to see today is that the brain and the spinal cord are arranged with big clusters of white matter and big clusters of gray matter. In the spinal cord, that gray matter has a butterfly shape. And it's found central, in the center of the spinal cord, and it's surrounded by white stuff. Now, because the gray butterfly, because it's gray, we know that it's primarily what? What part is going to be gray? Cell bodies and dendrites, right? And even the axon terminals, right? The very ends are also non-myelinated. So you're thinking, okay, the gray butterfly region of the spinal cord must be largely cell bodies, dendrites, and the axon terminals. The white matter around the outside, which is broken up into areas called funiculi or columns, would be therefore largely myelinated axons. That myelin is being produced by what kind of cells? Oligodendrocytes here because, right, the myelin's made by oligodendrocytes. We're in the central nervous system, right? If we were out in the peripheral, then the myelin would be made instead by Schwann cells. Okay. So we're thinking about this gray butterfly surrounded by white stuff, and the gray butterfly is broken up into areas called horns. Now, those horns, right, there's a dorsal horn, there's a lateral horn, and there's a ventral horn. And you can replace the words posterior and anterior instead. Those horns, right, so here we are, anterior horns, lateral horns, posterior horns, and then the other section would be the strap that connects the two butterfly wings together, and that's the gray commissure. In the very center of that gray commissure, there is a central canal, isn't there? Now, so we've got the white matter and the gray matter of the spinal cord, And where I want to jump to is that beautiful colored image that I know you're going to stare at. Pretty easy to label here. Okay, I hope this looks really familiar to you, that you're comfortable with the parts, just the roots and the funiculi and the horn regions. But the image I want to stop at just for another moment is this one, because I think it's such a rich slide. So this slide shows us a lot of things. It shows us the gray butterfly. It shows us, right, but the butterfly here has been replaced by colored regions. And those colored regions represent the 4 divisions of the nervous system. There are, coming in your spinal nerves, 4 different types of fibers. Remember, fiber here equals what? Fiber in the nervous system equals an axon, right? Fibers. An axon equals a nerve fiber. So, and what is a nerve? A nerve is a bundle of nerve fibers, right? So it's a bundle of axons. Some of these axons coming through your spinal nerves would be somatic sensory. What kind of information are they carrying? Somatic sensory is the stuff coming sensory more from the surface of your body, from your skin, from your muscles, your joints. That's the incoming information. We would call that also afferent.
So those are with the sensory only.
Sensory only, but within the spinal nerve, right? So these are sensory neurons. So the information traveling through those individual neurons is unidirectional, from the outside toward the spinal cord. Now, I'm also going to jump ahead. Remember, each region of your body is divided up into dermatomes, segments of skin. So in other words, each of these spinal nerves is bringing in somatic sensory information from a strip of your surface body. Okay? Then there's also visceral sensory neurons. Those visceral sensory neurons are bringing information in from your viscera, coming in from your gut, from your heart, right? From all of your organs, deep, visceral sensory things. Now remember those two blue ones, they are the afferent, and they're traveling from the outside in, aren't they? And this information always travels in and goes to the posterior root, and there we see the posterior root ganglion. What's in that ganglion? The cell bodies for these very sensory neurons. So that bulge is it's a bulge of cell bodies, right? A cluster of cell bodies in the peripheral nervous system is a ganglion. And that information's coming in. It's going to synapse with cell bodies of interneurons. Cell bodies, gray. And so where are these synapses happening? In the gray butterfly. Because what do we have in the gray butterfly? The very ends of these axons, terminals, gray, going to dendrites, gray, going to soma, gray, of the interneurons. The interneurons are very, very short. They don't have a lot of axon, therefore they don't have a lot of white myelin. And then they connect to cell bodies of the outgoing motor neurons. So again, the gray butterfly is largely gray. There's very little axon in that area. And then it's going to synapse with the outgoing neurons, 2 types of motor neurons. The yellow ones are the autonomic motor neurons. Where are they going? Autonomic neurons are going to go to smooth muscle, cardiac muscle, or gland, things over which we have no direct voluntary control, automatic. Versus somatic motor neurons are going to head to our skeletal muscle, our voluntary muscle. So again, we have all 4 of those pieces of the nervous system traveling through these spinal nerves. The outgoing information, remember, is coming out through the anterior root. So everything in the anterior root is motor, whereas everything in the posterior root is sensory information. What's another word for a cluster of gray matter inside the central nervous system? A cluster of gray matter in the peripheral nervous system was a ganglion, right? A cluster of cell bodies, gray matter. A cluster of cell bodies, gray matter within the central nervous system is called a nucleus, right? Nuclei. So these two horn areas, right, are motor nuclei, aren't they? They're gray clusters surrounded by white stuff. And the two blue horn areas would be considered sensory nuclei. What do you think? Go back. We're at minute marker 20. Okay? So again, I told you yesterday we were around 60, weren't we? 60, 69 when I went through this. I would just suggest if you're going to go back in and listen, just go back and listen to that little tiny area. This is a very rich slide, and if you hear it a couple times, it'll sink in. It really, really will. So we went through the nerves. We then went through the idea of spinal nerves, and each spinal nerve is carrying all of this different kind of information that I just described. So that includes here the spinal nerve when it comes out. Think motor. The motor nerve when it comes out is going to split. It's going to go through the anterior ramus. There's also going to be some in the posterior ramus. If it's outward, where is this posterior ramus going? This motor information is going to do what? Cause the muscles of your back to move, right? Because that's the motor of your muscles. The incoming information, though, is going to do what? Come in from your skin, right? The sensations of your back. And that brings us to the idea of dermatomes, these segments of skin. Each area of your body is assigned to a different cranial nerve. All except C1 are giving you sensations on your body. When you get to the cranial nerves, you're going to see that cranial nerve number 5 gives you sensation to your face. That's the trigeminal nerve. But every other part of your body is coming from one of your 30 spinal nerves, except, again, no C1. C1 just isn't involved with sensation on your body. Spinal cord injury, again, if there's a complete breakage at around C6, what would be intact? What was not affected at C6 that we talked about? The diaphragm. Because the diaphragm was innervated by the phrenic nerve, and the phrase is C3, C4, C5, keep the diaphragm alive. So if there's a break at C6, then everything above that is intact. There's no problem with breathing. However, you will have lost the use of your arms and legs, so you are a quadriplegic, but you have complete respiratory control. If instead that damage was up around C4, then you would have some partial issue, wouldn't you, with your respiration. If instead your break was down in the thoracic region, then your arms would be spared, but you would still be a paraplegic without use of your legs. A person at T6 would have issues with control across their abdominal region, so they might tend to slouch more in the wheelchair, whereas a person down at L1 would have very good core support. They'd have no problem sitting up, if you will, pretty well, but they would still have no use of their legs, so they'd still be a paraplegic. It does, yeah. I mean, clearly, remember, these nerves are not only sending out muscle control and receiving sensory information, but they're also, each spinal nerve is receiving signals responsible for defecation. And your defecation reflexes are largely in the spinal cord. So damage to the spinal cord would leave someone unable to control defecation, or urination, they're going to be on in a bag and have a catheter, unable to control those processes. Remember that if you have damage to half of the body, here the gray, the lighter color, is the area of paralysis. So if you had damage to just your legs, you're considered paraplegic, damage to all 4 limbs, a quad, and damage to half the body, hemiplegic, which would be evident of a brain injury. These two are evidence of a spinal cord injury, and this would be more evidence of a brain injury if you're hemiplegic. A group of nerves traveling together is a plexus, and there are 4 plexuses that we've discussed in lab. The intercostal nerves do not form a plexus because they are separated already by the ribs. And then we went through the 4 plexuses, right? So you can identify the plexus. These extra slides were just sort of an FYI, just to kind of bring home the overall idea that each nerve is responsible for sensations from a restricted area of your body and sends motor instructions also to each area of your body. What we're really not describing here, though, very much, is that these nerves also control or receive visceral responses and send visceral autonomic responses. So you could have issues with your internal organs as well. And we got through the 4 plexuses last time. We got through the conversation about reflexes, and I think we're about to the point where we need to slow down and start picking this up.
Are the reflexes a large portion of the test?
Not a large portion, no. I mean, just understand that a reflex is an involuntary, rapid response. It does not involve the brain. It is happening, it's controlled, if you will, by the level of the spinal cord. It happens faster because it doesn't involve the brain, right? It happens faster, and these are thought to be protective mechanisms in place. It's sometimes hard to imagine just how fast this happens. I mean, reflexes happen before you're even aware of them. I mean, that they're happening before that moment. So I think yesterday you asked, well, if I, swat a fly on my shoulder, that's really not so much reflexive, right? You actually made the choice to use your right arm to cross over your body to hit your left shoulder to hit that fly. So even though that was really, really fast and very annoying, that's still not so much reflexes. You still controlled that issue. Reflexes, we're talking about that really sudden, oh my goodness, I touched something hot, I stepped on something, I stubbed my toe, things we wouldn't really anticipate and things we would never do to ourselves usually, right? If you know it's coming, right, if you know you're going to stab yourself with a needle, you can kind of flinch, right, and kind of, in a way, minimize the reflexive. But by definition, a reflex cannot be controlled, right? That's the basic definition. We know there's some wiggle room in there that it is possible to sort of train yourself or to anticipate. Clearly, a diabetic is not flinching and saying, ouch, every time they stick themselves with a needle. At some point, you sort of get beyond that in anticipation. But typically, reflexes are going to be something you would not be able to suppress. Okay, so this idea of monosynaptic, so let me go back. I think this is about where we were on Monday. So ipsilateral versus contralateral tells us what? Ipsilateral means on the same side of the body, contralateral on the opposite side. Ipsilateral, if I stick my finger out with my right hand and burn it and pull back my right arm, that's all happening on the same side of the body. That would be an ipsilateral response. If I instead, as an example in lab, stepped on something sharp with my right foot, but then involved my left leg to protect me, to catch my weight, it's crossed over to the other side of the body, so it would involve some contralateral signals. The simplest of all reflexes is considered a monosynaptic reflex. One synapse, monosynapse. Well, up until now, whenever I've described a synapse in lab or here, I always said there were 5 things involved, and the third thing on that list was always an interneuron. I was lying a little bit. Monosynaptic reflexes do not involve an interneuron. Instead, there's a sensory neuron coming in, and it synapses directly onto a motor neuron that goes out. One synapse, right? If there's an interneuron involved, then there's at least 2 synapses. So monosynaptic reflexes, very, very quick, the most basic of all of your reflexes, and the best example of this is the patellar knee-jerk reflex that we've all experienced when having a physical. Now, polysynaptic reflexes involve at least one interneuron, maybe more, right? But at least one interneuron, therefore a minimum of 2 synapses. The more synapses, the slower it is, but it's still amazingly fast, right? So even a polysynaptic, even a complex polysynaptic reflex is faster than having to wait for the signal to go to the brain and send the signals back down to move. Okay, so it's all relative as far as speed here. It is taking a little bit longer, but still really fast. Now, just take a look at this. On the left is a monosynaptic reflex. Again, your patellar knee jerk. So the physician, the person hits the patellar ligament, and As a result, the quadriceps femoris muscles sense that they've been stretched unexpectedly. And when skeletal muscle is stretched unexpectedly in reflex, the muscle will contract. And so as the quadriceps femoris muscles contracts, the leg extends and it kicks out. So what's happening is that when you hit that ligament, signal comes in. Oh, there it is, right? Now what kind of signal is this? It is afferent. Would you consider this sensory motor or visceral sensory? It's coming in from the surface muscle and joints, isn't it? So this would be coming in as a good old somatic sensory neuron. Comes in. Oh, there it is, right? There is that unipolar cell body that we see within the ganglion. Comes in the back door and doesn't even touch us, it doesn't go to an interneuron. Instead, it goes directly to a motor neuron on the way out, and then comes out the anterior root and goes to, in this case, the organ. Now, what kind of signal is this? It's going to what kind of muscle? It's going to skeletal muscle, right? Your quadriceps femoris muscles are skeletal. So this would be a somatic motor neuron. So we think of our voluntary muscles as being completely voluntary, but that kind of gets washed out when you think about reflexes, right? Because reflexes are the involuntary movement of your voluntary muscles, right?
I have a question.
Yes.
Do they use their own, like it looks like, pretty, like the arc. Do they use their own arcs or can they use other, channels that?
This is very simplistic. You're struggling with what I struggle with. To teach this information, you have to bring it down to basically simple cartoons like this. But what's missing here? What's missing? Do you ever become aware that you just kicked your leg out? Yeah, I mean, after someone hits you kick your leg out and you look down, you're aware of it, right? You know you did it. So what's missing in all these circuits? Information going up the spinal cord to the brain saying, hey, dummy, you just moved your leg. Right? So or when you burn yourself, right? You reflexively pull back. And that's all I talk about here. But just a moment later, a signal went up to your brain and you sensed pain, and then that signal came down and you said, ouch. Right? So clearly these circuits are way oversimplified. What's missing is the complexity of other signals in your nervous system informing you that you just got injured or just got stretched or just did your reflex.
Would a reflex, I don't even know what to call it, use its own channel like that, or because they're unipolar, right, when they're going up?
Right, that's a sensory neuron. They're all unipolar.
Okay, so, but obviously those are different because you have no awareness that you're doing it. Are they recycling basically the same synapses?
That same receptor up here could also sense something that was done voluntarily.
Okay, so they're the same thing, just they
used in different ways.
So there's something that differentiates what they call the voltage that comes through.
The amount of stress, the amount of stimulus.
Okay.
Yes.
So they are basically the same axons.
Yep.
And synapses.
Used
but
in a
they
different
just know
way.
that they're different.
Right.
Okay.
Now, on the right-hand side is a polysynaptic reflex. Same idea, right? Here you're being burned by something. You reflexively, right, the signal comes in, you move your hand away. Again, there's that sensory neuron. But now what's it doing? It's synapsing with at least one interneuron. And so therefore it's a polysynaptic. There's two or more synapses. And then the signal goes out. Okay? Again, very, very fast. Now, this kind of completes the idea that you were saying. How do your muscles know that they're being stretched? So when you hit the hammer on the patellar ligament, patellar tendon region, and you get this reflex, it happens because within the muscle there are sensors. And these sensors are sensing stretch. So there are these stretch reflexes, and that's actually what's initiating this. So when you tap on that tendon, what does the muscle feel? That it's being stretched. And that stretch sensation is what triggers the sensory neuron to initiate the reflex. So they're actually here within the muscle. Now, if I say muscle, the first thing that comes to my mind is motor. Do you agree? Right, that when I say muscle, up until now, you've only heard about muscles being the receivers of motor neurons. And when the signal goes to the muscle, it moves. Right, that's really all we've talked about. But I want you to realize that muscles are also sensory. Look at this. Within the muscle, there are actually sensory neurons. What are they doing? Sensing stretch. So your muscles themselves are sending signals to your brain saying, my muscle is straight, my muscle is 90 degrees, my muscle is flexed, my muscle is relaxed. So you've got constant communication. It's all under the radar, right? You don't think about it. But you have signals going from your muscles. So muscles are not only receiving motor information, but they're also sending sensory information. Okay? And that's exactly what's going on. So when the muscle gets stretched, those sensory signals are sent. Now, one of these reflexes is referred to as the Golgi tendon reflex. You would not want your muscles to be excessively stretched, because what could the muscle do? You could pull so hard that the tendon would separate from the bone, and this is not what you want, right? You would never want to pull the muscle so hard that you cause damage to the tendon or connection to the bone. And so there are safeguards in place. And these are called the Golgi tendon reflexes, or the Golgi tendon organs that control the Golgi tendon reflex. Now, I know it's confusing because this is the same name as the Golgi complex within the cell that distributes proteins. Same spelling. What's that? Same guy, yeah. Kind of famous guy, right? So here, let me show you what these Golgi tendon reflexes or organs are. At the interface of the muscle and the tendon, right? We recognize that. There are these sensory neurons. So which way are they heading? They're outgoing, right? They're afferent. And if this tendon starts getting stretched too much, then this neuron is going to send a signal. It sends it to the spinal cord and tells this muscle here to do what? What would you tell that muscle to do? Relax or contract? Relax, right? Because to contract any further would maybe continue the damage. So these sensors are sitting right there on the interface of the muscle and the tendon. And if this muscle starts getting stretched too much, this signal goes back and involuntarily tells this muscle to relax. Have you ever watched any of the Olympic type shows, the He-Man shows, and there's a guy in competition? Now, in the gym, he was used to lifting 750 pounds, right? Whatever, right? And all of a sudden, he's going for the gold, and he's now lifting 775 pounds, and he's never lifted that much before, but he's going to go for the gold. And you watch these guys, right? They're massive, and they're about to do a deadlift, and it's like their body turns into spaghetti. It's not because they didn't have the mental desire to lift that weight, but their own Golgi tendon reflexes just kicked in, and their own muscles were sensing that they were being stretched beyond their limit. And that signal reflexively was sent to their muscles saying, relax. And they drop that weight. It's like they become a noodle. And it's reflexive. They can't fight it, right? This is not something you can suppress. So when this signal is sent, you don't have control over it. You don't have, you don't have control to overcome it. Your muscles are going to go limp.
So how does there become the injury?
Well, injury can still occur, right? Absolutely, you can still tear tendons, right? If the injury is sudden and there was no opportunity to pull away from it, right, then clearly you could tear the tendon or have an injury. But if you're ramping up to it, if you will, and you're getting to a threshold, there's the opportunity to save the damage. But clearly we can't protect everything, right? So pain really is that same kind of thing, right? You would think, who's going to break a bone? Before the bone breaks, there's pain. But if there wasn't time, or the trauma was so great, then who cares about the pain? You're gonna break the bone.
Um, when, when that happens and the tendon is broken, does that Golgi tendon cease working?
I don't know. Honestly, I don't know if when the tendon is repaired, if there's a successful repair, I would imagine that those reflexes would be minimized. That any damage to the tendon would also damage some of these reflexive neurons.
He did, my dad, was in an arm wrestling contest and, it went in his shoulder.
Did some damage to the tendon,
Yeah,
rotator cuff areas, and then,
I mean, he's got a scar about that wide, I don't know.
and now,
He had a stroke, so he doesn't have any, like, feeling on this side.
but
So,
between the damage and the stroke?
did he have pain, is that what you're saying?
Or did he have any less reflexes or control in that
Yeah,
shoulder?
I mean, I wasn't born then.
Okay,
so
okay.
I don't know, but I just was just curious, because he's actually stronger on the one side now than he was before, but he doesn't know it because he can't feel it.
Got it.
So,
Yep.
if that makes any
Got it.
sense.
It does. It does. So when you go into the doctor for a normal checkup, they may check a couple of your reflexes, right? They'll do the patellar tendon reflex. And what are they looking for? They're looking to make sure there's no spinal cord injuries, and you haven't complained about any neuromuscular issues, but they're still going to kind of check it. If you were to go in after some trauma, accident, fall off a ladder, they're going to test your reflexes from head to toe. And again, what they're looking for is there any damage potentially to your nervous system. So if you just fell, you're going to be looking for a hypoactive, right? Some sort of damage that just occurred. Your reflexes can be normal, yay. They can be hypo or hyper. So they can be understimulated or they can be overstimulated. And if they're hypoactive, then it does suggest damage to the spinal cord in the short term, or maybe there's a muscle disorder, right? Such that when you hit the muscles, they don't respond because there's something wrong with the muscles themselves. But if you just fell off a ladder, they're worried about spinal cord injury when they're doing your reflexes. Your reflexes can also be hyperactive, suggesting that there's damage to the brain and that there are signals being sent like an epileptic or something where there's some tremors, and that would be something wrong, not with the spinal cord per se, but more with the brain. There can also be some spinal cord injuries with hyperactive as well. Not to memorize this list, but what muscles are you testing when you do the patellar knee jerk reflex? Where are you? Front of the leg. And you know that which group of nerves are going down the front of the leg? Lumbar, right? The lumbar plexus, those nerves are going down the front of the leg. So when you're doing the patellar knee jerk reflex, you're actually testing, are there any problems with the lumbar nerves or therefore the lumbar spinal cord? So if the patellar knee jerk reflex was a problem, then there might be issues at L2 to L4 area because the quadriceps femoris muscles are being innervated by nerves L2 through L4. There's a whole series of other tests you could do all the way from the plantar reflex, sliding something across the bottom of your foot. That would be sacral, all the way up, right? Sacral, lumbar, thoracic, cervical. So there are reflexes that one can do all the way up the body to test for different regions of damage. And the only one we're describing here is the patellar knee jerk. But you see why we do it and why a clinician would go through a whole series of these. A neurologist would go through a whole series of these in the clinic to assess any damage to the spinal cord.
I thought the spinal cord ended at L1.
It does. But there's a lumbar region,
Okay, so
and there's a thoracic region, and there's a sacral region. So again, that whole idea, yes, the spinal cord ends at L1 as far as vertebra go, but there's still a lumbar region and a sacral region way up high. And so if you had problems with your sacral nerves, there could be problems at the sacral spinal cord. Problems with your lumbar nerves at your lumbar spinal cord. So when I showed you this image way back here about spinal cord injury, when I'm saying damage to L1, well, okay, what if I had said damage to L5? That might be a better way, because this is all within the spinal cord proper, but what if I said there was damage at L5? What do I mean by L5? there's a lumbar region of the spinal cord way up around L1, right? That's called the lumbar region. And if there was just the very tip of the lumbar part of the spinal cord damaged, low, L5, you would lose your sacral nerves and the coccygeal nerve. But you would have L1, L2, L3, L4 intact. That's still way up there around L1, isn't it? Okay, see what I'm saying? So we're talking about the region of the spinal cord proper. The spinal cord itself does end at L1, but it has regions along the entire thing from cervical down to coccygeal. The cauda equina would be what's coming off from the lumbar region and traveling down through the vertebral foramina and then comes out at its appropriate place along the vertebra. Okay, so a nerve that's coming down, a nerve that is coming out right here, let's call that L4, right? Where did that nerve come from? Inside here, there is cauda equina, and the spinal cord itself ended around L1, didn't it? So that nerve, L4, originates up here, doesn't it? It originates up here in the lumbar region of the spinal cord, up in this area. So that ends the spinal cord chapter. Any other thoughts about spinal cord? That leaves me with about an hour and 20 minutes to get through some of the brain section, and I don't have to get all the way to the end since I've already pre-recorded the end of this next discussion, but I'm hoping to get there so that I can walk you through it together. So moving on to what you have as chapter 13, the chapter on the brain. Some of this is repeat because I'm going to once again describe the meninges. There's still the dura and the arachnoid and the pia mater around the brain, just like there is around the spinal cord. So some of this is going to be repeated, and what you're going to focus on in this chapter are going to be the different regions of the brain. Some more, again, more anatomy. Look at this picture and tell me why it's poorly labeled. Why did I do a horrible job when I put CNS on this title? What should this thing really say? It really should just say the nervous system, right? Because the CNS is only what? The brain and the spinal cord. But this picture also has, the dissection includes what? I've also got on here the cranial nerves that we'll talk about and the spinal nerves, right? And those would be part of the PNS, right? Not the CNS. So that's why this is, not well labeled, okay? Because the cranial nerves and the spinal nerves are part of the PNS. Now, what can we see here? What is this group of nerves, right, about here? And not the right color. What's this group of nerves right about through here? Looks like a cluster of nerves, doesn't it? It looks like it might be going off to the arm. So that's part of the brachial plexus, isn't it? Do you see the cluster? See a clustering right here? Going off to the arm. So that'd be part of that brachial plexus. Up here, these nerves would be part of the cervical plexus. And again, remember the thoracic nerves don't have a plexus because they're separated by the ribs.
It just looked like his neck was pretty long.
Yeah, because you're looking at the back, aren't you? So what is this little area? What is this area right here? That rounded area is the, you may recognize that? I'm getting kind of cluttered up there. Do you agree that, where would the occipital bone be? Right about here? Right? Skull? So what would this region right here be? What the spinal cord attaches to what? Medulla oblongata. And then there's a sort of an oval-shaped region. That would be the pons. Okay? I wouldn't have you label it here, but you've seen those a little bit in lab. But they were on sheep brains, a little different looking. Okay. Okay, so this chapter is about the brain and about the cranial nerves at the end. Your brain's about, for guys, about 3.5 pounds. Women, smaller brains. Men do have bigger brains. But the sad news is that size of brain does not equate to intelligence. Okay? So Neanderthal man actually had a bigger brain than man as far as size. And what determines your intellect is instead the number of active synapses. This is why we get grandma playing cards and doing, any kind of mental games, because if we can keep the brain active and keep the synapses going, that maintains their overall well-being. It's the same reason why baby Einstein, right? Why we have babies in utero listening to music and being stimulated, right? Kids who are more stimulated before and after birth, we know make more active synapses and do better in school because their intelligence is higher. And kids who have been neglected don't get those opportunities, and so their number of synapses is lower, and so they're at a deficit throughout life in their overall functioning and in their intelligence. So again, about 3.5 pounds, kind of a jello-like feel to it. You felt those sheep brains, but they were pickled for a little while. They're not quite the same, but the brain would be a little bit more jello-like. It's sitting in a fluid-filled area, the cranium, and that fluid around it is the CSF. And the brain is an absolute oxygen hog. It is requiring and getting about 1/4 of all of your oxygen. So it must have a continuous supply of oxygen. It must have a continuous supply of nutrients. Without it, you're in trouble really, really fast. So the brain cannot go without oxygen. Just a few minutes can leave someone with significant brain damage. Now, the next conversation should look really, really, really familiar, because just like the spinal cord is protected by bones and by meninges, so too the brain is. Except here, the bony portion is the cranium. You know it as the frontal bones and the parietal bones and all the bones of the skull and of the cranium. And there are also the meninges, same ones. They're continuous around the brain and down around the spinal cord. And also there is CSF, made by the same old cells, the ependymal cells. And there's one more thing that helps to protect the brain, and that is the blood-brain barrier. You may have heard about this, but there is basically a more stringent control of what is able to enter into your brain tissues through the bloodstream. And that extra level of control is referred to as the blood-brain barrier, and it is maintained by the astrocytes. Remember when I described the astrocytes in the picture? They were those green star-shaped cells, and they were creating structure within the brain and the nervous system, but they also had their little feet-like structures wrapped around the capillaries, because they are regulating what can and cannot come into the brain. So the astrocytes are maintaining the blood-brain barrier. So the names are the same, the location's different. Here we'd call them the cranial meninges. In the last chapter, I referred to them as the spinal meninges, but it's the same old thing. You've got the dura, the arachnoid, and the pia mater. They are protecting the brain. They're providing space for fluid. They're providing a space for the blood vessels to also be protected. What good is it to protect just the brain if you're not protecting the blood vessels, bringing oxygen and nutrients also to the brain? So we better be able to protect all of that. And so we do that through the meninges. And you know them, right? The PAD from inside to out, the pia, the arachnoid, and the dura. So let me go through pictures of these. Now there's one slight variation on the theme here. Around the brain, the dura is actually 2 layers. Now in the spinal cord it's 1. But around the brain, the dura is composed of 2 layers. They're both tough layers. Together they're really tough. And these 2 layers are called the periosteal layer and the meningeal layer. Okay, what do their names suggest? The periosteal layer sounds like it's associated next to the bone, right? Perioste. And the other layer must be closer, if you will, to the brain or to the meninges. And so that's a deeper layer. I'll show you a picture of these. Now, normally, these 2 layers of the dura are fused together, and you would not see them as 2. But there are a few places where these 2 layers separate, and where they separate, they fill up with blood, and these blood-filled areas are called the dural venous sinuses. dural, they're in the dura. Venous refers to blue blood, blood that has already gone to the brain and is coming back down. It's blue because it's less oxygenated. And sinus because where it separates, it creates a space. So these dural, venous, sinuses. I'll show you a picture of all of it in a moment. Let's go on to the arachnoid mater. Nothing here you don't already know. It is directly deep to the dura. It is composed of a delicate web of fibers. And that's where we get the name, the spider web looking appearance. Those are collagen and elastic fibers. And collectively, those fibers create a trabeculae. Now, where have we seen that word trabeculae before? Spongy bone. Remember in spongy bone, that webby, trabeculae, that network, that webbiness on the inside of bone? Same idea. These fibers are creating a trabeculae within the arachnoid mater. Just below that, right, between the arachnoid and the dura, there is that subdural space. We labeled that with the spinal cord as well. And then below the arachnoid is the subarachnoid space. Now remind me, what is in that subarachnoid space? That's where the CSF is, right? So underneath it, in that spidery web area, that's where the fluid is. And then we go down to the pia mater. Again, it's the innermost shrink-wrapped layer to the surface of the brain. It is going to follow every contour of the brain. Now, you know those contours as gyri and sulci, right? So every noodle is a gyrus, every groove is a sulcus, and that pia mater is actually shrink-wrapped right down and following every little groove and contour on the surface of the brain. So this picture does okay. There's a couple of things in here I want to point out to you. It's not as quite a high a picture as another one I'll show you in a moment. Here's the dura. Now here the dura is not shown as a double layer. It's just shown as a single layer, right? The light green. The darker green is the arachnoid. Just below that are the spidery-looking fibers, the collagen and the elastin. That's filled with fluid, isn't it? But look what else that fluid's doing. It's also protecting a whole network of blood vessels. Right, so there are blood vessels also being protected in these layers. Like I said, what good is it to protect the brain if you're not also protecting the oxygen and nutrient supply going to and from the brain? Then the pia is shown here in pink, okay, shrink wrapped. And notice how the pia dives down in every little groove right around the brain. Now this picture also introduces something. Notice that I'm going to draw this big area over here on the left. That area would be the meaty part of the brain. And I want you to see that there are two layers in here. And it says up here, gray matter. So the gray matter is this area here. And the pink would therefore be, quote, the white matter. Now, how is that different from what we just saw in the spinal cord? In the spinal cord, the gray matter was where? In the center, the gray butterfly, surrounded by white. What we're going to see is that in the brain, the outer layer is gray. And the majority of the inside of the brain, the medulla, is going to be white. What makes the outer layer gray? Same reason, cell bodies. What makes the medulla, the center stuff, white? It's primarily axons, surrounded by myelin, made by oligodendrocytes. Okay? Now, in bone, what did we call the outer layer? We called it compact bone. There was another word, though, used too, Nancy, and that was cortical. Cortical bone. And cortical referring to the cortex. So the outside of the brain is referred to as the cortex. You labeled that in lab, but you labeled 1,000 things in lab last week. So the cortex was the outer layer of the brain. It's gray. The cortex of the brain is gray because it contains all the cell bodies. The center of the brain is white, again, because of axons. And we call the inside the what? the medulla, or the medullary area. Remember in bone, we had the medullary cavity, and outside could be called compact or cortical bone, cortex and medulla. The brain, outside layer, cortex, inside, medulla. In the kidney, we're going to see the outside, cortex, inside, medulla. In the ovary, we're going to see cortex and medulla. These are generic terms. They're used across all anatomy for solid organs. So there'll always be the outer layer called the cortex, the inner area called the medullary or the medulla.
If the top layer is gray and they have the axon or the heads or the body,
Cell bodies, yes.
so on the top, would that mean that the axons are going straight, and then wouldn't they have an unmyelinated center?
You're going to see a beautiful picture in just a moment. Let me try to, it's really complex, but there's a beautiful picture coming up that I think is going to help clarify how this architecture works. Okay? Now, here's the cartoon that I like a little bit better because it shows, this one shows that the dura is actually 2 layers. So let's go through all this. So we have the skin on the scalp, right? Very top layer. Then underneath that we have the bone. This is flat bone, isn't it? This is bone of like the frontal bone or the parietal bone. So we know this is flat bone, and flat bone is characterized as having periosteum on both the outside and the inside. So underneath your skin, there'll be a layer of periosteum. There it is. Then your flat bone. And what's on the deep side of the flat bone? Another layer of periosteum. Well, that periosteum is also, AKA, known as the periosteal layer of the dura. So it's the same thing. It's like that idea of hypodermis and superficial fascia. We've got 2 different names, and we realize it's the same thing once we put it all together. Then deep to that periosteal layer is the meningeal layer, and together that's the dura. So the dura here is shown as this double layer. Now, most of the time, these 2 layers are fused together, and you really wouldn't tell them apart. But there are these places where they separate. And in those separated areas are blue-filled sinuses, and that's a dural venous sinus. It's blue blood coming back from the brain, and it happens to collect in these sinuses. Now, this picture does a nice job of showing you that subarachnoid space. You can see those webby fibers in here and the fluid-filled areas. This figure also does, I think, a better job of showing you gray versus white, right? So the gray layer around the outside, basically all the gyri would be, if you cut into them, gray, right? The outside layer of the brain. And then deep to that would be the white medulla, or the white medullary area. Let's talk about the area, the anatomy of the brain. You know most of this. There's a couple things on here that are new for you. The brain can be broken up each hemisphere, right, can be broken up into 5 lobes. So there are 2 frontal lobes, 2 parietal lobes, 2 temporal, 2 occipital, and don't forget there are 2 insulas that are deep inside the brain. So each hemisphere has 5 lobes. There's also, and collectively, all of those lobes are referred to as the cerebrum. And then deep in the brain is what's called the diencephalon. Now, when I see the word, when I imagine the diencephalon, I imagine a peach when I think of the brain. And the fleshy part of the peach are the lobes, right? The fleshy lobes. And the pit of the peach is the diencephalon. So the diencephalon is the core, the pit, the center of the brain. Now, in the diencephalon is where you find the thalamus and the hypothalamus. So maybe now you can picture it, even before we get to the picture, that core center kind of oval-shaped region inside, deep in the brain. There's also the area called the epithalamus here, and the epithalamus is where the pineal gland is. Then we get to the brainstem. The brainstem can be broken up into areas called the pons and the medulla. You saw that in your lab quiz as well. I had those little areas. I had, I said the pons and the medulla, and collectively the blank was, what is that? That is the brain stem. And then don't forget, you all can recognize pretty easily the cerebellum. So those are the major structures, the major parts of the brain when we look at it as a whole organ. Of course, we can break the brain up into 2 halves, 2 hemispheres. And those two hemispheres, again, are broken up into these functional areas. And I'll talk a little bit about what each area of your brain is responsible for doing. On the surface of the brain, you can easily identify, I know you can, many gyri, the noodles, and the little grooves between them, the sulci. And, as we'll see, coming out of the brain, there are 12 pairs of cranial nerves. Coming out of the spinal cord, we saw 31 pairs of spinal nerves. So we've got 31 pairs of spinal nerves popping out of the spine, and now we have 12 pairs of cranial nerves popping out of the cranium, out of the brain. So I've already mentioned this. I've already mentioned, prepared you for this. Okay. So what makes gray matter gray? Again, I keep saying it. Gray matter is gray because it lacks much myelin. So that's going to be your cell bodies. That's going to be your dendrites, the axon terminals, and some unmyelinated axons. Remember, there isn't as much white around some of those unmyelinated axons. So collectively, this would be gray. Whereas white matter in the brain is going to be is mostly composed of the myelinated axons. Do you recall that your brain, from the very beginning when we were talking about the tissues, your brain was formed from your ectoderm? That is, from the outer layer. So when you were that little ball of cells and that little gastrula, and you started having 3 layers of germ layers, the ectoderm, the outer layer, becomes your epidermis as well as your nervous system. So that's where it all is derived from, is the outer layer. And those outer layers maintain their identity as the cortex. So again, that idea of the outside is the cortex. So your cerebellum has a cortex, your cerebrum has a cortex. And we've already seen this as well, that the arrangement of gray and white is different in the spinal cord than it is in the brain. Again, in the brain, what do we see? The white matter is deep to the gray. That's opposite of the spinal cord. In the spinal cord, we saw the gray butterfly deep to the white. And we know what makes the white matter white: myelin. And myelin is found around the axons. And a bundle of axons in the central nervous system we know is called a tract. So what's going up and down the spinal cord and what's traveling through the brain are not nerves. Remember, nerves are only found outside in the periphery, the spinal nerves, the peripheral nerves, the cranial nerves. But within the white of the spinal cord, within the white of the brain, we have bundles of axons traveling together as we call a tract. And then, do you think the whole middle of the brain is white? No, there are islands of gray. Oh, here we go. And those islands of gray would be what? Islands of cell bodies. And each of those has a name, and we're not going to learn them. There's hundreds of them, right? But here's that word again, nucleus. So what's a nucleus? A nucleus or a nuclei, a cerebral nuclei would be what? A cluster of gray matter somewhere within the central nervous system. So within the big old medulla, within the big white center of the brain, I'm going to assure you there are little clusters, little islands of gray matter, and each of those little clusters would be called a nucleus. Some of them, they all have names. They all have something they're doing special, and we'll deal with more of those next semester.
Are those the areas where they take the pictures, when they see the...
A little bit. You'll hear about the basal nucleus. You'll hear about different structures within the brain, and you'll hear that word nucleus a lot. So hold on to that. That'll be a focus in your very first lab in 106. You'll do some neuroanatomy, and you'll look at a number of parts of the brain, and you'll learn a lot more about the brain next semester. So we've already talked about nuclei versus ganglion, right? Yes? And we've already talked about nerve versus tract. Or nerve versus tract, right? And we've already talked about Schwann cells versus oligodendrocytes. So remember, those were those 3 distinct terms that were different if we were dealing with the peripheral or the central nervous system. Just make sure you're clean with those. So you've already done this in lab. This should be pretty comfortable. So we know that the entire brain, if it's in your palm, we can divide the whole brain up into two hemispheres, left and right, separated by a very deep groove. That deep groove is the longitudinal fissure. It separates the left from the right hemisphere. What we can't see, if I went deep down, way down in that longitudinal fissure, there would be, and I'm going to make it white, just to make it white, but down in here, there would be a band of white matter that connects the left to the right hemisphere, and that would be the corpus callosum. And I think that is a typo, and I apologize, that should be CA on callosum. So that's the corpus callosum, and that would be what connects the two hemispheres. We know that the surface of the brain is filled with these bumps called gyri, and the grooves between them called sulci. You can't appreciate it here on this image, but there is a central sulcus. The central sulcus is the groove, the official divider between the frontal lobe and the parietal lobes, between F and P. You'll see it in a colored brain in a moment, and I'd only ever have you label it on a colored brain. There's also a lateral fissure, which we can't see here, that separates the temporal from both the frontal and parietal lobes. So when I turn this to the side, you'll see that better. Now we can see this groove right here. That separation, that would be the central gyrus. And it's separating, it's the groove that separates the frontal lobe from the parietal lobe. There's also going to be the transverse, the lateral sulcus, and this is the lateral sulcus here. And what is it doing? It's separating the temporal lobe from both the frontal and the parietal lobes. Cerebellum down here. I showed you the same slide in lab, and I just want to mention some of the major functions of your lobes. The frontal lobe is the area of your brain involved with motor control. So when you choose to move your body voluntarily, those instructions are coming from your frontal lobe, unless it's a reflex, right? And then we don't have control over it. But all of your voluntary controlled movements are initiated from your frontal lobe. So a person who has a stroke or damage to their frontal lobe might have problems with movement. So a person after stroke who has any sort of paralysis, you can know with great certainty that it's part of the frontal lobe that got destroyed in that stroke. Now there is a crossing over, so that damage to the right frontal lobe would cause potentially parts of paralysis to the left part of the body and vice versa. Okay. This is also the area of the brain involved with your inhibitions and your intentions. And so individuals with damage here may also have issues with appropriate behavior, appropriate language, right? Grandma, who was an angel, now swears like a trooper after a stroke. She has suffered something in the frontal lobe.
Would you call that discretionary as well?
Discretionary, yeah. Discretion, lack of discretion, they start doing very inappropriate things. You know, when someone just kind of rants on and on and on, and they're just inappropriate, my first thing is, okay, something went on with the frontal lobe at some point, right? Could have been long before you were born, but there's something not connected quite right in the frontal lobe.
It's very difficult to deal with that too.
Very difficult. There are only some people in the world meant to deal with them, and they are thankfully the nurses and doctors who work with these patients. But it's not.
Yeah,
It's not for me either, right? Not for me. So that's your frontal lobe. Then there's the parietal lobe. Damage to the parietal lobe is going to give you difficulty perceiving what's going on around you. So you may have damage to your parietal lobe. You're moving around fine. There's no paralysis. There's no movement issues, but you're a little bit clueless. You're just not picking up on the signals being sent to you. You may not understand instructions very well. You may not, you just are not connected to the world because most of your sensory information is going up to your parietal lobe. The temporal lobe, easy enough, on the side of the head, language centers and hearing. A person with damage to the to the temporal lobe might have issues with speech or understanding speech, either one. And I'll give you some more specific examples of that in a moment. And then the occipital lobe is primarily involved with vision. Okay? So remember your eyes are up here, right? Oops. Your eyes are up here, but that information is going to travel back to your occipital lobe. And so we would say that your occipital lobe contains your, listen to this word, visual cortex. Because where is that this is the optic nerve, right? We know the optic nerve is the nerve coming out of the back of the eye. So the optic nerve is carrying that information from the eye, and it's going back to the occipital lobe. Where on the occipital lobe? To the cortex of the occipital lobe. Because what's in the cortex? Cell bodies. So the synapse, right? The information, your visual information is going to travel from your optic nerve, go back, become the optic tract. Yes? You agree to that? The optic nerve is sitting on your eye. It's in the periphery. When that optic nerve goes deep into your brain tissue, now we're going to call that the optic tract. And the optic tract is going to synapse where? On the cortex, on the cell bodies of the occipital lobe. So we would say that the occipital lobe contains the visual cortex. Here's where I'm going to talk a little bit more about parts of the brain. Just a couple of extra things here. So sometimes it's difficult to get your bearings when there's no cerebellum. When there's a cerebellum on the brain, no problem. You know that that's back toward the occipital lobe, and the frontal lobe is as far away from the cerebellum as you can get. But when the cerebellum's gone, sometimes I have difficulty figuring this out. What you want to look for, and I'll go back, is you want to look for this indentation. Okay? So if we look forward now, here is that indentation. Oops. Right here. So this would be the frontal lobe up here. And what is this groove called? This groove was the lateral sulcus. If I pull down on the lateral sulcus, pull down on the temporal lobe, and pull up on the frontal lobe, what's hiding back there? That's the insula. Right? So if I pull down on the temporal and pull up on the frontal lobe, if I separate at the lateral sulcus, I would get a peek of another whole area back there called the insula, that insulated lobe. Do you agree that this is the frontal lobe? And that this must be the parietal, and back in here would be the occipital lobe, right? Cerebellum would be here. It's missing in this picture. There is a guitar pick area right here that I want you to see, and it's called the, let me move that if I can, it's called the Wernicke's area. Okay, so this area right in here, Wernicke's area, kind of looks like a guitar pick to me. Now, Wernicke's area is a very particular part of the brain, and it is involved with understanding language. If someone has damage to this particular region of the brain, they're going to have difficulty understanding, perceiving, taking instructions. What lobe am I in? Kind of at the interface of the temporal and parietal. And what hemisphere am I on? Is this left or right? This is your left hemisphere. Now, your left and right brain are not the same. Your left and right brain are not mirror images of each other. There are certain functions that are only found on one side of your brain and not on the other. And this is one of them. So Wernicke's area is typically only on the left hemisphere. Now there are those people who are completely backwards, and they're great studies. They're great research institutions who study individuals whose kind of brain is completely backwards, if you will. But for most people, the Wernicke's area is on the left side of the brain. There's another area here that I want to point out. It's not marked quite as well, and that's this bluish purple area. And I just messed that up, but this area over here, let me get rid of that. That's not working well. Just this bluish area right here. And that's referred to as Broca's area, or what's called the motor speech area. A person who has damage to that area, and what lobe are we in now? Frontal. Person with damage to the Broca's area would definitely have difficulty speaking. So they wouldn't be able to articulate, they can't get the coordination of their tongue and their throat and the muscles of their mouth to coordinate the activity of speech. Again, we're in the frontal lobe. Motor movement, intentional movement. So if you have damage to the frontal lobe, you may have body difficulty with all sorts of paralysis. But if specifically you had damage to the left hemisphere, to this area called Broca's area, you would have problems speaking after a stroke, okay, after that brain damage. Two other things that I want to point out on this brain are the blue and yellow stripes. So right here, this blue, now what do we call a noodle? This is the whole noodle, that would be a gyrus. And this is the last gyrus of the frontal lobe. It's the most posterior of the frontal lobe. And then I want you to look at this yellow strip. That is also a gyrus. And what is in between them, what separates that blue from that yellow, is the central sulcus. So now where am I? That means that that blue is the last of the frontal lobe, and the yellow is the beginning of the parietal lobe. And I'm telling you this, it would not be easy to identify if it were not a fully colored model or figure. I'd be hard-pressed to find the central sulcus on any of the brain models upstairs. You might be very hard-pressed to find it in an actual brain specimen. But there, of course, there's always going to be a boundary somewhere. And here it's very easy to see in these cartoons. So that groove in between is called the central sulcus. Okay? So the central sulcus. The other name for this, and you're going to see it in the next slide, and it tells you over here, that last little strip is called the precentral gyrus. Because where is it? Right before the central sulcus. And then the yellow strip is also called the post-central gyrus, because it's the groove right after the central sulcus. Okay? What we're going to do here is look at them from a different view. And I think this one is so much, they're both good images to label. They're not too busy. So if I look at this image, again, I have no cerebellum. I've kind of lost my perspective here. And it tells me, thankfully, that this lighter orange is the frontal lobe. Okay? And the darker salmony color is the parietal lobe, and the gray is the occipital. The temporal's too low and off the side, I can't see it. The big groove up to down would be what? That's my longitudinal fissure. But now, what is this? That divider would be the central sulcus. Again, the strip right in front of it as part of the frontal lobe is called the precentral gyrus. The strip behind it in the parietal lobe is the post-central gyrus. Okay? So that's what's important from this slide. Remember, too, your dividing, if you will, left and right. You're looking down on the brain, aren't you? So this is one of the few times that right is right. Do you agree? You're looking down on the brain. So right is on the right, you're looking down. Typically, when you're looking at a model, you got to flip left and right, don't you? But in this case, do you get your perspective? You're looking down on the body, so right, your right is the right of the brain. Now what I'm going to do is I'm going to take this slice of material right here. Okay, I'm going to slice that out. Pretend this is the birthday cake. And I'm going to take a slice. And I'm going to take this slice, I'm going to go deep into this gyrus. And I'm going to take that little slice of cake out and I'm going to lay it on its side. And there's my slice of cake. Okay, and let me give you your bearings here. We're looking at, maybe I should have done the other side, but we're looking at a slice. There would be another half of this over here just to give you your bearings. And this is just that little slice. It's called the precentral gyrus. The other name for this is the motor strip. What lobe are we in? We're in the frontal lobe. And I told you the frontal lobe is responsible for your motor movement, your voluntary movement. The motor strip is this really cool strip of land in your brain, and this little guy has been stretched across the motor strip. And he's been stretched out to represent the number of motor neurons going to different regions of your body. What proportion of your motor strip is designated to controlling this part of your body? So what we see is that there's a really large number of motor neurons going to your face. That's going to include the sense of, the movement of your face, all the complex movements of your face. Also, the movements of your jaw and your tongue and your swallowing, all that stuff. There's also a fair number of motor neurons going to the fine motor control of your hand, but not as many motor neurons going to the rest of your leg and your foot. Okay? And this strip of land is literally spread out around this precentral gyrus. Now, what side of the brain is this? If we... Yeah. It doesn't even matter to me. I'm losing my perspective here as I stand up here. But this, let's say this is the right side of the brain. This motor strip would be controlling the what? Left side of the body. Okay, so I just want you to remember that the motor strip is controlling the other side of the body. What is this little indentation right here? Just to give you perspective. If this is the frontal lobe, then this would be the temporal lobe, and that indentation would be the lateral sulcus or fissure. You'll see it both. And what would this represent back here? Yeah, that's the insula, right? It's like another little lobe inside, just to give you perspective. And what would this little what would this little white matter be right there? That's the corpus callosum. And what would this deep groove be right here? The longitudinal fissure, because there's another half of the brain over here, isn't there? So are we getting it? We're kind of getting our perspective where we are with this little slice of the body. Now, the number of motor neurons equals the size of this little guy that's stretched around. So that's the precentral gyrus, and that's what's shown over here. This is the same thing. This is the motor strip, or the motor cortex. That's where we were. It's just a different guy stretched out, but it's the same exact representation. And that blue strip up here then is the frontal lobe. That's the motor strip. On the right-hand side, the orange strip is actually the sensory strip. We're in the post-central gyrus, and this is called the sensory strip. Not the motor strip, but the sensory strip. Remember I told you the parietal lobe is involved in receiving your sensory signals. And just like there's a motor strip sending signals out, there's a sensory strip receiving information. And the same idea, there's a person stretched out over this sensory strip to tell us areas of the body which are more sensitive, that receive more sensory neurons. And it makes sense because isn't our hand incredibly sensitive, right? To touch, fine touch, all sorts of things. And isn't our face also extremely sensitive, right? Our face, our tongue, all that. So pretty much, it's kind of a mirror image. It kind of is a mirror image. The only difference is that on the sensory side, you've got the genitals, and there's no genitals on the motor side. Other than that, it's kind of a mirror image. There's about the parts of our body that have more motor control also happen to be the areas of our body that have more sensory sensation, right? More sensations. So on the left, what do we have it? We have the precentral gyrus. We're in the frontal lobe, and it's involved with motor control. On the right, we have the postcentral gyrus. We're in the parietal lobe, and we're receiving upcoming sensory information to this strip of land. So what if your stroke was up here? We're in the frontal lobe. See that little circle up there? If your stroke was way up here in the frontal lobe, would you have any paralysis? No, but you'd be acting inappropriate. Whereas if your frontal lobe stroke involved the motor strip, you might act appropriate, but you would have issues with some paralysis. So again, we're seeing that different parts of the brain have different functions, and if there's damage to those areas, we can expect different symptoms. And the way we figured this out is that over the years, right, people sustained head injuries. 50 years ago, we didn't have MRIs. 30 years ago, I don't know exactly when, but when we were able to start mapping the brain and seeing areas of damage, not from the outside, but from the inside, we would say, okay, that person just had a stroke or a head injury, and it damaged this little part of the brain, and then we look at how they're different. Oh, that little part of the brain must do that. And we've been mapping this for years. So now, right, when a patient has a series of symptoms, we can pretty well say, they've sustained damage to this part of the brain, and the MRI will back us up because this is pretty well mapped.
But nothing you can really do about it.
Nothing you can really do about it. That's the unfortunate part, right? So we can take a peek. Oh, look, you've sustained damage to your right frontal lobe, specifically to the motor strip. So you're going to have paralysis on the left side of the brain. The greater the damage, the more paralysis. Can you expect to get some recovery? Some. It won't be magnificent, but there'll be some recovery, and the patient will go through lots of physical therapy. And within the first 6 months is where most of the improvement will be seen, if there's going to be any. After 6 months to a year, we've kind of maxed out any kind of plasticity or recovery that can be expected. That's the general rule of thumb that I've been told. So any kind of improvement you're going to see is usually going to be rather quick, within the first months after the damage. After that, what you're left with is your residual function, and that's what you're left with for your life. Any questions about motor strip, sensory strip? Falling into place a little bit? Now, the next part, I'm going to throw some new words at you, but they all have the same root. All that's different is the prefix. So that's what you're focusing on. All these parts of the brain have the term in them, encephalon. Well, encephalitis, right? Inflammation of the brain. So all these encephalon words are parts of the brain. And before it is a prefix. So there's the proencephalon, the mesencephalon, and the rhombencephalon. Not that difficult. And I'm not going to harp on this too much on the exam. The part that I really, really, really want you to know is going to be the telencephalon and the diencephalon. And I'll expect those two words to be part of your vocabulary. The other ones, to be honest with you, I'm not going to test you directly on. Okay, so focus on telencephalon and diencephalon. Now let me break this down for you and then show you some pretty pictures and make the sense of this. Your cerebrum, right? What did I say? The cerebrum is what? Your lobes and that pit, that core, that nugget in the center. The brain, the lobes, are also called the telencephalon. Now, what's tele? Telodendria, telophase. telencephalon. So tele means last or end. This is the last part of your brain to form, the telencephalon. Physically, your frontal lobe is the last part of your brain to form. And I think I mentioned in passing that the telencephalon, right, the frontal lobe specifically, is the last part of your brain to get myelinated, leading to very weird decision-making skills of young people because they don't yet have that intentional area well developed, and it's not yet fully myelinated, and young people make silly mistakes, right? They do silly things without thinking. What was I thinking? Well, okay, son, you don't have your frontal lobe's not quite made there yet. Okay, yeah. Okay? And unfortunately, it's not made till we're 25, and it starts to break down by the time we're 30. So there's about 5 good years in there, folks. So if you're not there yet, and look forward to it, and once you're hitting 30, it's all downhill. So just kidding. Well, we'll talk about how the brain does start to show signs of aging around age 30.
I think they should give that information to parents at birth because I've shared that with a lot of people and they all go,
Oh.
I'm like, no, it makes sense.
Oh, it all makes
And
sense.
I'm like, yeah, there really is something. to
Yeah,
it.
and the insurance companies know it, because when does car insurance get cheaper?
Yeah,
25.
Yeah.
There's a reason.
In car rental.
This data makes sense. Okay? And some other people know it. It's just a big secret. Okay, so the brain, the cerebrum, right? It's the last part of our brain to be developed. Now, not only is it the last part of our brain to develop in you and me individually, but from an evolutionary standpoint, cockroaches have a brainstem, right? Simple animals have brainstems. We're on the upper end of the evolutionary chain, and therefore we're the last, if you will, to have developed. And so you can also think of it that way. So the fleshy, beautiful cerebrum that we have as humans is called the telencephalon. In the center of that, that was the pit, the core, the center, that is the diencephalon. I used that word earlier. And that's where the thalamus is. That pit, the core of the peach. So again, if we're using the peach analogy, the fleshy, edible part is the telencephalon, and the pit is the diencephalon. Then there's a very small part of the brain called the mesencephalon. What does mes mean? Middle, right? So it's the middle part. Okay, so again, those prefixes are going to help us. So the mesencephalon is the middle part of the brain. It's very, very small, really nothing I'll have you label. And then there's the rhombencephalon. It must kind of be rhomboid in shape. And within this area is the brainstem. And this is where you have the pons and the cerebellum and the medulla oblongata. And yes, there are other names for those, but I told you I'm not going to quiz you on those. So don't spend any brain power memorizing metencephalon or myelencephalon because I'm not going to ask you about them. Okay? So let's put all this together and figure this out. In this image, the big blue circle is all of the fleshy cerebrum. We would call that, right, we could call it the, what? Telencephalon. Deep in this, not visible from the outside, but deep in the core of the brain would be the what? Diencephalon. And collectively, that's your forebrain, right? Your forebrain is your telencephalon and your diencephalon. Then the midbrain, that mesencephalon, that little middle area that we really can't see, is, I'm just pointing to it. It's just this little kind of connecting area, nothing to really identify. Then the red is circling what's called the hindbrain, and that's going to include the cerebellum, the pons, and the medulla oblongata, right? What you would call the brain stem, really, is in a common language. So a cockroach has a brain stem. I mean, simple animals. An earthworm has basically a basic nervous system made up of things like medulla, pons, and cerebellum. Only we, mammals, right, have a well-developed proencephalon, have a well-developed telencephalon. So I'm going to start this little movement through the brain from the most primitive to the most advanced. You're not going to memorize everything on these lists, just to appreciate that the most primitive structures of our brain, I should say back, the most primitive activities that we do, breathing, heart rate, our basic living functions, the respiration rate, all that's going to be happening where? In your brain, in your brainstem. Because the brainstem is involuntary. We don't have control over our cerebellum, right, and our medulla and our pons are part of the involuntary part of our brain. For us to become conscious of something, it has to go up into our cerebrum. It has to reach our frontal lobe and our parietal lobe and our lobes for it to become conscious. So the unconscious things are going to be down in the brain stem. This is going to be your pons, the medulla oblongata, and the most basic of living things like, darn it, I was off, I don't know how long. I'm not going to quiz you on these terms here, but in the very bottom, I know yours is just a black spot, right? But here, the red on here is black on yours. That would be the very medulla oblongata, right? That's the very base. The spinal cord's coming up and connecting to the medulla. And again, this is where your most primitive things, don't memorize this list, but things like the control of your blood pressure, the control of your respiration rate is traveling up through this area, okay? So most primitive areas.
This is the part I got wrong on the quiz in lab. So does the stem end at the bottom of the medulla, or that's where the spinal cord begins?
So yeah, so in this picture, what would be right here? Occipital bone.
Okay.
Okay?
Yep.
So we'll just say anything, and I should put it right here, really, okay? Okay, there's your occipital bone. So anything outside the foramen magnum would be spinal cord. And anything up in this area we're going to call brain stem. Okay? We're moving up. Now we're at the metencephalon. Again, don't worry about the name here, but this is the pons and the cerebellum. Again, what we're looking at are very primitive things. You're looking at your control of breathing, heart rate, pressure, bladder control, basic life functions, things that we don't have control over directly, right? And then the cerebellum is the largest part of this. Now, the cerebellum has a very important part in your movement and in your balance and your posture, but involuntary. Okay, this is involuntary movement. Your cerebellum is not sending instructions for you to move your arm, but the cerebellum is going to help coordinate that movement. So when I look at a dancer, an athlete, when I look at a figure skater, and they're doing this beautiful athletic move, my first thought is, they have a beautiful cerebellum. Because the cerebellum helps to coordinate your muscle movements, makes you more fluid. Damage to the cerebellum would make you more jerky in your motions. You would still have control over the voluntary decision-making to move your arm, but if you had damage to your cerebellum, that movement may not be as smooth. Right? So a person with beautiful grace and athleticism, not only do they have a well-tuned, muscular system, but they also have the gift of a well-tuned, if you will, cerebellum, because it controls your involuntary movement, balance, posture.
Could the pons be responsible for people with like anxiety and panic?
There can be some of that, but we're going to see, not this semester, but next semester, there's a part of the brain called the amygdala, which is your emotional centers deep in the brain, and that's where we think about the emotional responses coming. So some of that, I won't call it imbalance, that's not sensitive enough, but some of those anxiety and some of those issues may be coming more from that part of the brain. So the cerebellum is not just this little structure hanging off the back of your brain. Actually, half of your brain's neurons are squeezed into that cerebellum. So what does that tell us? It's pretty important, right? It's pretty awesome, pretty important stuff. And it is attached to the, the base of the brain, as you know, back here. There's your pons in front of it, the medulla oblongata sitting next to it, right? And again, it's coordinating your movement. Problems here would result in ataxia. Ataxia would be an unsmooth physical movement, a jerky movement, an uncoordinated state. We would say you're ataxic. Okay? You have ataxia. Short story, my wife's aunt started experiencing some balance issues, and it just kind of happened on really fast. And couldn't figure out what it was. And finally, after about, I forget how many months, but way too many months of looking and trying to figure out what was going on, they finally noticed in an MRI a mini stroke that had occurred in her cerebellum. Now, when you have a stroke today, if you take an MRI tomorrow, the tissue has not yet completely died. So you have to wait sometimes a while for the dead tissue to appear. And it was so microscopic, they kind of missed it, and they ended up going to Mayo, and they found, again, you can't do anything about it, but at least now we have a diagnosis. So her uncoordinated state, her dizziness, was a result of a slight, relatively small, minor stroke in her cerebellum, which left her a little bit ataxic, a little bit unsmooth, a little bit out of balance sometimes, and gave her quite a few problems, actually. Now, just look at other brains. These are all mammals, right? And here's our beautiful brain. An elephant has a huge cerebellum. Look at that. Again, size doesn't necessarily mean intelligence. So the elephant is not smarter than us, I don't think, right? We've got a bigger brain, but it's not smarter than we are. And even little, the overall structure, we looked at sheep brains, kind of look like a miniature cat brain down here, doesn't it? So remember the cerebellum's on the back side because the spinal cord goes down, straight down the back, right? So a little bit different architecture. But again, all these mammals have a nice cerebellum and then a brain stem, they all have a nice cerebrum, and they all have a brain stem with a spinal cord coming off from it. Let's keep moving up. Mesencephalon. This is that middle, right? Mesentery, in the digestive lab, the mesentery was the layer of structure that kind of connects all the intestines together in the middle. When you think about your small intestine in your fetal pig, all the intestines were kind of bound together by mesentery. So mes in the middle. So the mesencephalon is the midbrain. Again, nothing here I want you to label or know, but notice it's still your basic structures. You're not yet up in the telencephalon. You're still down in the brain stem areas. Now there is an area down here that you may have heard of, and that is the substantia nigra. And I'm just going to point it out because you may have heard about it. The substantia nigra is a portion or an area within the midbrain, and this is the part of the brain that starts to break down with Parkinson's disease. So if you know anyone with Parkinson's disease, it's a degenerative muscle control disease. And here, again, like the cerebellum, there's control of movement. These individuals start getting tremors, and it's because there isn't enough neurotransmitter made. But you may hear of the substantia nigra, especially with Michael J. Fox and other individuals who are really big into Parkinson's research. And that is this little area of the brain. So there's something that's gone wrong there. It could be brain damage, or it could just be there are certain cells in here that start to break down, leading to Parkinson's. Now, we're almost there. We're in the diencephalon now. We're up in that nugget, that core, that pit of the peach. And here in the diencephalon is where you find the thalamus. Can you picture it? The thalamus in the brain was that center. If you looked at the half-brain model, the thalamus was that big kind of pit circle in the center. The corpus callosum was kind of right above it. So right below the corpus callosum is the thalamus area. And also in here is the hypothalamus, that little triangular region below the thalamus. And what was hanging off the bottom of the hypothalamus? The other P. Pituitary. Right? So the pituitary gland, that little P-shaped gland, is hanging from the bottom of the hypothalamus. So you can picture it. Show you a picture in a second. And then there's also an area of the diencephalon called the epithalamus. So it must be what? On the thalamus. So we have the thalamus. We got under the thalamus, hypothalamus, and we have on the thalamus, the epithalamus. It's all part of the diencephalon. So here it is, right, in this cartoon image. What is this white area here? I'm coloring in the what? That's the corpus callosum. So we're looking underneath the corpus callosum, right? And you don't see the pituitary here, but that whole red area would be combined the thalamus, the hypothalamus. Now, don't worry about this. Not worry about the basal ganglia. So the thalamus, the hypothalamus, and what else did I tell you was in here? The epithalamus. So let's look at what we've actually seen. Now, this brain's been a little bit squished for the PowerPoint stretch, but again, what do we have here? This is the corpus callosum, right? We recognize that band of tissue here. Kind of underneath it, where this red circle is, that's all the thalamus. I'm going to clean this off. There's kind of a triangular region right there. That would be the hypothalamus, and what's missing, but right here hanging, would be the pituitary gland. Back here on the thalamus is a gland called the pineal gland. I told you it didn't show up in our models upstairs very well. It is an endocrine organ. We'll talk about it in the future, but that is also part of the diencephalon. Now, let's quiz everything else on here. What is this area? No, it's labeled, but this oval-shaped area is the pons. This oblong region below it is the medulla oblongata. The occipital bone would be here, so everything distal to that would be the spinal cord. And then there's the big old cerebellum. And when I look at it, I can see that beautiful arrangement called the arbor vitae.
Is there anything that you can do to strengthen certain areas of your brain, such as your...
Anything you do to strengthen certain areas of your brain. The people who make Luminosity want you to think so, right? The little brain games. Clearly we can, I mean, there's a lot of neuroscience that suggests that music and, again, stimulation to young people can stimulate more synapses, that as you're learning, you're creating new synapses, right? Being a lifelong student, maintaining new activities helps to create new synapses. You know, listening to music, doing crossword puzzles, all those things are all thought to maintain and try to keep up the number of active synapses. I don't know if there's anything we can do specifically. I mean, some of us are just blessed with certain gifts that, you know, we certainly didn't ask for or haven't developed, but we just have them, and other things we don't have.
No matter how strong it is, if you have a stroke, it wipes out, it wipes out.
Yeah.
Right? I mean, it doesn't,
All,
strength of it doesn't really,
well,
I wanna say matter, but.
true, but like if you're talking about, like, you know, movement of your arm, right? I mean, you could have a stroke that affects just a portion of the motor strip. So you might have partial paralysis, but still have use of the arm, but maybe not total paralysis or not total use. If you had a stroke that wiped out that entire portion of the motor strip, then you would have essentially no use of that arm. So again, the extent of the damage and the area has a lot to do with it. Yes, damage is damage, it's gone, but how much area is gone? And are there other areas nearby that can sort of fill in the gaps? Remember I told you that damage to the spinal cord, if you damage just C3, or if you damage just C5, right, you still have some phrenic nerve function. Same idea with the brain. It's really hard to say that little pinpoint area only does that. There's a lot of cooperativity in the brain. So when you lose a portion of the brain, there can be other areas that can kind of, quote, come to the rescue or fill in a loss of function. And again, that's normally going to happen within the first 6 months to 12 months after injury. And some neuroscience would suggest there are other things you could do to facilitate that, but it's limited. It is limited. Okay, we're at the last part of the brain. This is the telencephalon. Where are we? We're now in the cerebrum. We're finally in the conscious part of your brain. You're up in your lobes. Okay, so the telencephalon, the last part, the most magnificent part of our brain. This concludes the hemispheres. This includes the cortex, right, the outer layer of the brain. This is going to include all of your magnificent human things like thought and voluntary motion, language, reasoning, and perception. I'm not suggesting to you that your dog doesn't have voluntary movement, right? Because they also have a telencephalon. But their overall thought processes and their language processes certainly aren't as developed as ours. So yes, mammals have movement, they have memory, they have perception and reasoning skills. Some dogs certainly more than others, right? Some animals more than others, and some humans more than others. But all of that magnificent, you know, human character, if you will, is housed here within the telencephalon. Now here's the coolest picture that I think will bring you back to what's going on with this white matter and gray matter in the brain. Before I go here, is there anything about from the primitive brainstem up through the levels that I need to re-clarify. Again, where does your consciousness start? In the cerebrum. Everything below the cerebrum, pons, medulla, cerebellum, the diencephalon, the thalamus, all that is your unconscious brain. You've got to get up through the thalamus to get to your consciousness. The thalamus is sometimes called the gateway to your consciousness. So let's see how that works. Look what we have here. On the left is an actual brain that they have teased all the matter, a lot of the tissue away. And what you're left with is all of these axon fibers. Well, what's an axon fiber? An axon is a nerve fiber, right? So it's sort of the same thing. So what you're seeing is coming up from the spinal cord. What's all this stuff? It's white, isn't it? And what's it doing? It's coming up, and almost every one of those fibers is going right through the center of what? What's right here? Thalamus, right? So you see that all that information coming up, if it's going to get up into our consciousness up in here, it's going to have to go up through the thalamus and into the diencephalon. Now, where are all of these axons going? Where are they all going? What's this outer layer called? The cortex. And what's in the cortex? Cell bodies. So where are all those axons going? They're all going to the cortex of a lobe, right? Because that's where the synapses happen, on the gray stuff, right? On the cell bodies and on the dendrites. Okay? So all of these fibers are going up. So that makes the majority of the brain what color? All this out here is going to be what? White. And it's going where? It's going to the cortex, where the cell bodies are. Now, what's the word we could use also for that? A bunch of cell bodies, right? Within the central nervous system. We could also call it a nucleus. Okay, again, a cluster of cell bodies, a cluster of gray matter would be a nucleus.
So are the gray areas in the white below the cortex, are those kind of stopping points then? I guess in my mind.
They are. They're
it's
relay
top to
stations.
bottom.
They're relay stations. So,
Okay.
so there would be, and I don't have them labeled here, but there would be areas. For example, there could be an area right here, or an area right here. Just making it for examples. A nucleus, a cluster of cell bodies. And what would that become? What I'm going to call a relay station. Because there might be some axons coming up, and they synapse to that. And then there'd be other neurons leaving that and going up to the cortex. Okay? So again, these nuclei would become relay stations. Right? White axon coming up, synapsing on cell bodies, and then another white axon going off to synapse somewhere else.
Is it because they have different functions that are necessary? Like, say they stop in that area in the frontal lobe and then continue on to the next lobe?
They're informing some part of your brain about something. So those neurons are bringing in information or sending information out.
Okay.
And and this is exactly where we're heading. Perfect. I got 5 minutes, and I think this is going to be perfect. But this gets to the idea of pathways, right? These neurons are moving up through your body with particular pathways. And these pathways are going to be carrying sensory and motor information, just like we've been discussing before. And where do these pathways travel? They're passing up through the white matter of the spinal cord, and we'd say up through the columns, up through the funiculi, right? And they're traveling up as tracks, right, through the spinal cord and the brain. So a pathway is going to consist of a tract and a nucleus. Break it down. A tract, again, is a nerve-like structure, bundle. And a nucleus would be where it's going, right? Because a nucleus is going to be a bunch of cell bodies. So that axon, those axons are going to synapse with a group of cell bodies known as a nucleus. And there are all sorts of nuclei in your body. So again, this idea of these little clusters of cell bodies somewhere found in the brain. And in lab, I mentioned the idea of ascending pathways and descending pathways. And I think now this is pretty straightforward. What are ascending pathways? Going up, they're carrying what kind of information? Sensory, right? Pretty obvious. And then descending pathways would be carrying instead motor. Where is that sensory information going? Not all of it, but largely going to the parietal lobe. And those descending pathways sending instructions are largely coming from the frontal lobe, the motor strip. Now, many of these pathways cross over. We talked about how damage to one side of the brain is seen on the other side of the brain. The big word for that crossing over is decussate. So these pathways are said to decussate. And over 90% of your pathways do cross over. They do decussate. There are a few that don't, but most of your pathways do cross over from the left side to the right side. Okay, so the last couple of things. The cerebral cortex is that, right? The cortex. This is, again, the most highest mental functioning area of your brain. This is where your consciousness, your memory, your reasoning skills, the things that make you largely human, because we have the most beautifully designed cerebral cortex of all the animals. Our cortex is largely here by the age 5. Kindergartners have huge heads. Right? They got huge little heads. Their brain's already 95% of adult size by age 5. So kindergartners, what's different, the size is there. It's not completely formed on the inside. We don't have all the synapses and we don't have all the myelination. But size-wise, a kindergartner has a huge brain, right? 95% of their adult size. Some of those neurons are not yet myelinated. All the neurons are there, though, right? The neurons are already formed. Because once you start losing neurons, remember, they don't get replaced. So we're not constantly making new neurons because neurons are not mitotic. Neurons are part of our development. They're formed, and that's it. And once they're formed, they're there. And if we don't use them, we don't use them. But once we lose them, they're gone. So all the neurons are there, just all the synapses are not yet made. And all of the myelin is not yet laid down in its proper way. I've mentioned MRIs. There are also PET scans. It was the last thing I mentioned in the very first lecture. PET scans use radioactivity to look for areas of the brain that are metabolically active or not. So a hot spot would be red, a blue spot would represent coldness, maybe not as much, and that's all computer enhanced. Here's a brain. I'm going to need about 5 extra minutes, guys, but I think it'd be worth it. The brain is hooked up to a PET scan, and in the lower left, a person is receiving auditory information. They're being spoken to. What area of the brain is lit up? Yeah, that's sort of Wernicke's area, isn't it? Wernicke's area was that little guitar pick area I showed you, was perceiving instructions, perceiving language. So that part of the brain is most lit up. This is the normal brain. And then over here, the information, they're receiving it. And then over here on number 3, it tells us that they are now responding to that information. They're speaking. So now what part of the brain is lit up? It's that area called Broca's area. That was the motor speech area. But also, can you imagine, where are we? If I could, I wish I could draw. I didn't have that skill. What strip is this? That lit up area is pretty much where? Motor strip. And where in the motor strip? If you look at that motor strip, what's on the side, down on the temporal lobe? What's on the side of the motor strip? The face, right? If you look back at that little stretched out man, his little face was down along the side of the brain, and then it went up to his hand and then around to his feet. So the side of the brain's all lit up. Specifically, right, that Broca's area is lit up, and we saw that it takes a lot of muscles, right, to articulate, to move the tongue, to move the mouth. A lot of the motor strip is lit up here as well. So it's cool. right? That we can actually see this functional imaging. As the person's actually doing something, we can follow what parts of the brain are lit up. Cool stuff. So again, on this picture, up here, that was the Wernicke's area. Over here is the motor speech area, the Broca's area, and you're right there also in that motor strip, aren't you? I told you that the left and the right brain are not the same. There are specialized purposes for each. Going back to this, what side of the brain is this again? This side of the brain, this is the left side of the brain. So again, your language centers, your Wernicke's and your Broca's areas are on, for the vast majority of the population, are going to be on the left side. So there are certain things on your left side, certain things on your right side. In fact, your brain is asymmetric in its shape. These asymmetric areas are called patalias, and your left brain is differently shaped from your right brain. And even more interesting, if you are right-handed, you have a difference in your right frontal lobe, and it's reversed if you're left-handed. So clearly, handedness is not just choice, right? There's truly proof that your brain is different. Let me just mention these two sides of the brain. They're called the hemispheres, and there are different specialized areas. Your left hemisphere is what's called the categorical hemisphere. And the reason is your left hemisphere is where your Wernicke's area was. We saw that. It's where your Broca's area is, your motor speech area. It's where most of your language skills are involved. It's also where your math and science areas are. So your analytical skills are more over on the left brain. Okay? So if you are left brain dominant, you're usually what-handedness? Right. So if you're right-handed, your left brain is controlling that dominance, right? So most people in the world are right-handed and are left hemisphere dominant. Not everyone. There are people too, there are people too who are left-handed, and what does that mean for them? Right? They're left-handed, their right hemisphere is their dominant hemisphere. What's going on in your right hemisphere? Well, this is your representational hemisphere, and this is the part of your brain that's involved with visual spatial relationships. This is your imagination centers. This is your musical and artistic skills. Okay, now, this is my perception on this, but I'm a saxophonist, and I'm a right-handed guy, very right-handed, and I'm a decent musician, but I never want to compete against a left-handed saxophonist. There's something about the left-handed people I know in my life. They have a musical skill that I don't have. And most of the left-handed people I know are artists. They're my psychiatrist friends. They're very bright, right? And they just have something about them that I don't have. And I think, you know, it's not 100%, right? But all my architect friends are left-handed. Right? Again, they have that creative side, they have that psychoanalysis side, they have that kind of warm fuzzy side that I don't have. I'm very, very analytical, right? That's my strength. I don't have that music. I mean, I have music, but I don't have the artsy stuff on my side. So you may think about that a little bit, and maybe some of the people you know. Now, people's lateralization is set by age 5 or 6. That is, by the time a kid goes to kindergarten, first grade, if they're going to be right or left-handed, is completely by that point firmly established. Now, people are ambidextrous, and they kind of live in the middle of this world. But if you look at a newborn, look at a 12-month-old, right? They're grabbing things with both hands. They're putting stuff in their mouth with both hands. They're not right or left-handed yet. And as they get older, you'll start seeing more and more progression toward right or left-handedness, such that by the time you get to kindergarten, it's pretty much set. Your brain has already been wired, and you're done. And the switching would be very, very difficult. Not impossible, but very, very difficult to switch. The last couple of things I have to show you, one more thing is about with the brain. Women have a thicker corpus callosum. What does that mean? They have more axons that travel from the left side to the right side and vice versa. So a woman who has brain injury has more connections to the other side of the brain to help make up the difference than a guy. So men typically don't do as well after a stroke because they have fewer connections from left to right. And that's just sort of a phenomenon that's real. But women have a thicker corpus callosum. You could argue women can multitask better too, right? But they have both sides of their brain in greater connection with each other than men. So not to memorize this at all, but I want to show you that the two halves of the brain are not the same. So over here on the right side, right, this is where you've got your, the left hemisphere, right? Your left hemisphere is where you have your rational thoughts and your language, and this is also where the motor speech area is. And then the right hemisphere has all of your musical ability, your spatial relationships, all of that good stuff. But again, differences left and right. All of your higher order processes, again, your language, your cognition, your memory, all of these are largely from your telencephalon, right? From your cerebrum. I got 3 slides to share with you, and then we're done, and I apologize for going over. The ventricles, I'm glad we did that spinal cord review, but it took up a little bit more time than I thought. So the ventricles are what? Ventricles are fluid-filled chambers inside your brain. Filled with what? CSF, same old stuff, right? Your brain is not only floating in CSF, but there's pockets of CSF inside your brain in these areas called ventricles. There are 4 ventricles in your brain. 2 of them are just called lateral ventricles. There's a left and right lateral ventricle. Then there's a 3rd ventricle and a 4th ventricle. Not hard to remember, right? 1, 2, 3, and 4. I'll show you a picture of those in a moment. Again, those ventricles are filled with CSF. And remember, who makes CSF? Yeah, the ependymal cells, right? So there they are at the bottom, ependymal cells. Don't worry about the word choroid plexus. Okay, don't worry about that term choroid plexus at all. Here's a picture. I got 2 pictures of your ventricles. From the side, this larger, that's one of your lateral ventricles. The other one's behind it, so you can't see it. So left and right laterals. In the very center, right in your diencephalon area, there's the 3rd ventricle, and the 4th ventricle is down by the pons and the cerebellum. Notice they're all connected by fluid, and there's the central canal of the spinal cord. So the central canal has the CSF, it goes up into the brain, fills up this ventricular fluid, and it's floating all over the place. From the front, it looks like ram's horns, doesn't it? Right? There's your 2 laterals, your 3rd ventricle, and your 4th ventricle. This picture shows you the same thing, just prettier colors. So again, 2 lateral ventricles, they're hiding on top of each other, the 3rd ventricle, the 4th ventricle, and again, continuous with the central canal. And they're all connected together. So this fluid's moving from one to the other. And how did the Romans get their water from town to the village? The aqueducts, right? And so too, we call these the aqueducts. The things that connect one ventricle to another are called the aqueducts, right? The water pathways. So the rest of this, come on. 2 slides on the blood-brain barrier. Again, I've already mentioned this. The blood-brain barrier is maintained by the astrocytes, and this is just another level of protection for the brain. It keeps the brain from getting harmful molecules from the bloodstream. So think of it as an extra level of just sentries of control. Again, controlled by the astrocytes that we've talked about in the past. Where would you have problems with the blood-brain barrier? I do want to mention this. I told you to cross off choroid plexus. But why would the hypothalamus and the pineal gland be an area where there is less blood-brain barrier? What are the hypothalamus and the pineal area both making? Hormones. Hormones must be released into the bloodstream. So the gate's open. Right? So if there are hormones being released from the brain into the bloodstream, that means the gate's open. That means that things can get in. So where would most things be able to get through the blood-brain barrier and somehow damage your brain? Where the gate's open. So near the hypothalamus and near the pineal gland. They're making hormones and dumping them into the blood, into your body's circulation. So too, viruses, pathogens, toxins could get into the brain at that place. Okay, the gate's open, things can get in. And there's your little astrocyte grabbing around the capillaries, maintaining the blood-brain barrier. What happens with the brain? About age 30, it starts to fall apart. Not horribly fast, But from age 30, you start seeing a decrease in the ability to maintain the brain's homeostasis, a decrease in the number of neurons, a decrease in the number of the amount of gray matter, the size, the removal of waste decreases, the synapses reduce, blood flow reduces, cognition reduces. Only thing that increases is the garbage, right? So the plaque, the crap that kind of builds up, starts to accumulate at age 30, and we know that that can accumulate to horrible levels and be called Alzheimer's in some pathologies. It used to be thought that Alzheimer's was sort of a natural progression of aging. We now don't call it that. It's a disease. We don't know what's triggering it, but it's not a normal progression. So people have dementia, that's normal. A little bit of forgetfulness is normal, but Alzheimer is another whole level of dementia. So last slide, you look at a person whose age, I forget the number, age here, let's say age 80, okay? And this was a collective MRI of all the brains in this study. And what you're seeing is that a normal elderly person is all blue, right? They have a beautiful blue brain here. And blue tells us that they have less than 2% loss. So yes, we start losing gray matter at age 30, but it's a very slow progression. So by age 75, 80, you still have the majority in a normal brain, the majority of your gray matter. Remember, gray matter equals what? Cell bodies equals synapses equals ability to perceive and send signals, right? Look at the Alzheimer's brain, a collection of Alzheimer's patients. What do you see? Splattering of different colors. Some areas with hot pink represents a loss of 30% of your neurons. So, and again, it's very sporadic. So you look at one Alzheimer's patient and their symptoms early on are very different. They start forgetting different things. There's just a different progression here. And again, it has to do with the randomness of what part of the brain has been damaged as to what's going to first be affected and what's going to later be progressive. Okay.
Get rid of the most recent synapses formed.
I don't think there's an inverse order, although it is interesting. From memory standpoints, some people will have brain injury of some sort, and they'll remember their childhood. And they may even remember a language that they spoke as a child that they haven't spoken in 50 years. So there is something about memory. Old memories are the last to forget, right? If that makes, did I say that right? So newer memories, short-term memory is lost first, and then people will still have memories as a childhood or, 50 years ago, but they can't remember you or anything in the last 25 years. So yes, it does appear that memory does kind of reverse in order of being laid down in your brain. I do apologize for going over. You guys have been very patient, but I think it's a good thing. I'm glad I was able to spend that extra time on the spinal cord review as we did that at the beginning. Listen to the recorded lecture. Right now I do have it chunked up into two little ones. It goes through the cranial nerves, and on Blackboard, under, I want to say assignments. Did anyone look at this yet?
I followed the link, but that might have been before you.
Yes, but what I want to point out to you is that on Blackboard, and let me just go ahead and get this on the video as well. Under, I want to say assignments, but that's what I want to check. Under lecture materials, and then under handouts. Nope, not there. Maybe it's under exam 3 materials. I'm losing it. Where'd it go? Oh, exam 4. Yes, thank you. Under exam 4. And cranial nerve preparation sheet. So open this thing up, and then there's also the answers right here. And it's just a little worksheet, but after you listen to the cranial nerve presentation, open this up. I mentioned it in the presentation. And it's a little story about what each cranial nerve is doing. And then the key is also posted. So you definitely want to look at this because on the exam, this is how I'm going to quiz you on the cranial nerves. I'm going to give you a little story, and after you listen to it, you're going to know that to smell something requires the first cranial nerve, and that to salivate requires the 7th and 9th nerve, and to shrug your shoulders takes the 11th nerve. So you're going to know that after you listen to that presentation. So read through this, then look at the answers, and let me know if there are any problems. And then I'll be glad to answer any questions about this via email over the weekend, or certainly at the beginning of the exam if there's any concerns at all. So again, I apologize, but have a wonderful 4th. Enjoy a little bit of freedom, but not too much, right?